So we are starting our next unit in biology with a look at modern genetics. So we're going from talking about how DNA stores information. We've talked about how that information is used to express itself and give us different traits, give every organism different traits, not just humans, right? Then we talked about how those traits can be passed down from offspring or from parent to offspring. Now we're going to talk about some of the more modern things that have gone on with genetics. Specifically, we're going to look at today the Human Genome Project of the 1990s through early 2000s, and we're going to look at DNA fingerprinting mechanically. In class, we'll talk about maybe some ethical dilemmas with DNA fingerprinting, but for now we're just going to stick with mechanics. As a reminder, we're now into the second half of the year. I expect you to be writing more of the things I'm talking about, not just the words on the screen, and that's going to start being reflected in the points that you earn on your notes, which is a homework grade. So, first, Human Genome Project. It started in 1990. It ran through 2003 with its initial run, and it had the big idea to reach for the stars and try to list every single base pair that makes up human DNA. Now, obviously, my DNA is different from your DNA. This is talking about the more general DNA that all humans share. But they also wanted to be like, this gene specifically codes for the color of your hair. This gene codes for your eyes. This gene says if you're going to have 6 fingers or 5 fingers. All right, that is the kind of idea and driving force behind this. They wanted to be able to use that information for practical uses, right? To make medicines, to understand human health better, to kind of give us the next big step in medicine. Unfortunately, they kind of ran into a problem in this, and the first problem was that some of the scientists working on it, because this was an international push, an international effort, some of them wanted to make money off of it. The other big group wanted to make this open source. They wanted to allow people free access to it. They kind of came to not blows, but they were arguing about it rather vehemently. It made the news. It made Time magazine in the year 2000, I believe. But the Bermuda Principles were where they got together and they said, okay, everybody working on the Human Genome Project, we got to get on the same page. This information is going to be shared internationally for free. The people who don't like that can leave. And that's what happened. All right. We're not going to push into the politics too much past what you guys are going to read about. But the complete sequence was published in 2003 at 99.99% accuracy. Now, I say complete sequence, but as you can see on the screen, that should have some quotes around it because it was really 92.1% complete. That was as far as science could get us. We just didn't have the technology yet. And for me, it feels weird because I remember 2003. For you guys, that's before you were born and seems like a long, long time ago. So, this is where they kind of stopped. Some people were still working on it, but the Human Genome Project itself was declared complete. The information that is in GenBank, right, is a sequence database of DNA. It's open access. It has nucleotide sequences and the proteins that are made from those. Right? It is produced and maintained by the National Center of Biotechnology Information as part of the International Nucleotide Sequence Database collaboration. And you can just go online and access it. If you Google GenBank, like I have it spelled here, it'll pop up like first Google search result with NIH, the National Institutes of Health. Right? That information was used for a lot of things. One of the big things that many of us might relate to is cancer identification. Many of you know people who have cancer or who've had cancer. We actually have a couple students right now as I'm recording this, who go to school who have cancer. And the tests we do to identify cancer cells versus regular healthy human cells, many of those tests are from the information brought out by the Human Genome Project, right? We also have some new cancer treatments that have come from that. Identifying and treating bacterial diseases is another big one. One of the ideas that they wanted to do was be able to use the information we gathered from the Human Genome Project to apply that to other things like bacteria. One of the things that's still being researched now is using bacteria to digest plastics so that they will biodegrade when they're treated with the bacteria, at least. So all sorts of things are coming from the Human Genome Project, and it's not just limited to humans. Now, recently, there was a kind of rush to try to get the other 7.9% of the human genome mapped. The winners of that competition were the Telomere to Telomere Consortium. They published in 2022, right? And now we're going to consider this gapless, right? Most of the sequencing that telomere to telomere did was near the telomeres, right? Telomeres are those long strings of molecules on the end of DNA that we think might be used for protection, might be used to make sure that the DNA doesn't get mutated or degraded during the replication process or transcription and translation. Right. One of the technologies that came out of the Human Genome Project is DNA fingerprinting. You can pause, write these steps down, and then click play, and I'll keep talking. All right, so DNA fingerprinting is very similar to the idea of our fingerprints and using them for identifying who we are. Since everybody's DNA is unique, we can use DNA to say, okay, this piece of flesh, skin, hair, eyelashes, saliva, whatever, right? It's got some DNA in it. We don't know whose it is, but we know some people who are in the area. We're going to take samples from them, and we're just going to match the two together, all right? So it's going to match using something like this, all right? And we're going to look at the banding patterns that you see at the bottom of the screen. To make those, we need to take the DNA, which is long double helix string, right? And we're going to cut it into chunks. Right, these chunks are going to be different sizes, but the restriction enzymes that we use to cut them are going to cut them at specific points for everybody. So everybody gets different chunks. And even if we have the same chunk, I might have more of those chunks than you do, specific at least. So we're going to take those and we're going to grow more of them in a polymerase chain reaction. That was the PCR initialism that you saw on the previous slide. Right? PCR is just going to grow a whole bunch more copies of that DNA. Then we take those DNA segments and we load them into a gel, which is kind of like Jell-O, but it's going to allow the DNA to move towards a positively charged cathode because DNA is slightly negatively charged. And the smaller the DNA segment is, the farther it's going to move towards that cathode. It actually also moves faster. Right. We're going to talk more about DNA fingerprinting and how it's used in solving crimes in class. Right. This is kind of our idea of mechanics more than anything else. All right. Remember, we're also going to talk about the ethics behind this. There's some ethical problems or concerns, at least, that some people have with DNA fingerprinting. So remember that I am starting to grade more on you writing down what I'm saying, not just the words that are on the screen. Keep that in mind. Make sure your notes are ready to go in class the next day. That way you can earn all the points for the lecture. If you've got questions, make sure to write them down. I at least want one question about the Human Genome Project and just a single opinion, one sentence, about do you think DNA fingerprinting is a good thing? And we'll see how that changes the more we learn about DNA fingerprinting.